Can everybody see my presentation as well?
Yes.
Okay. Pleasant morning to everybody. I am delighted to be serving as one of the guest speakers for today's webinar. And I think I should acknowledge Mr. Chris Argamino for including me in his quiz. Well, not related to me, it's the course, okay? And one of the popular bands in Ireland. Quite interesting because I bumped into them in one of the popular bars in Dublin. So very, very nice band. It's actually one of my favorite bands. Uh, Also, I think I'm quite fortunate because Dr. Lacerna presented before me, and Dr. Lacerna did an excellent presentation on essentials of metabolomics, which are also employed, okay, in metabolomics, clinical metabolomics. So that is going to work to my advantage, okay, because I don't have to discuss them in greater detail. So, but nonetheless, I'm going to mention them as part of my talk this morning. Okay. So now for this presentation, I'm going to talk about the application of metabolomics in the clinical setting. Hence, I, I entitled my talk as Metabolomics, bridging, bridging basic research and clinical medicine. This will be the outline of my presentation. So initially, I'm going to discuss an overview on metabolomics. I know you've had a lot of, you've got an overdose or quite practically saturated with metabolomics, and you pretty much know the definition of metabolomics at this stage. So I'm going to reiterate that as part of my presentation. And just earlier, Dr. Lacerna had this brilliant presentation on the various tools and strategies in metabolomics. In fact, Dr. Lacerna highlighted the typical workflow on untargeted metabolomics. And I will also go past, discuss it very quickly because it will form important part of my talk this morning. And the highlight of my talk would be the application of metabolomics in the clinical setting. And toward the end, I shall discuss the applications, my research initiatives, my personal experience in metabolomics, specifically with emphasis on applying this field in studying the complement of metabolites in this biological fluid, the breast milk obtained from the Filipino lactating women. And lastly, I'm going to present the summary of my talk. So I suppose you're very familiar with Oliver Fien at this stage. Okay, so Oliver Fien is considered the father of metabolomics. Okay, and he proposed a lot of developments in metabolomics. In fact, he has over 200 publications to date in metabolomics. He now serves as a professor in UC Davis and is also the current director in a very well-known metabolomics research center, Analytical Services and Metabolomics in US, the NIH West Coast Metabolomics Center. Now, by definition, metabolomics involves or refers to the screening of small molecules that we call metabolites that are present in biological samples or biological fluids. Now, you may wonder how small these molecules are. Okay, so generically, these molecules must be less than 1,500 Daltons. Okay, now the complement of these metabolites, or the inventory of these metabolites is what you call the metabolome of the organism. Therefore, the metabolome of this organism can provide a snapshot of the metabolism. And this is also widely recognized as the biochemical or the molecular fingerprint of the organism. Hence, the metabolites or the inventory of metabolites in an organism can provide the biological and health state of an organism. And this is practically where my presentation this morning will revolve around. Now, metabolites comprise an enormous chemical diversity of compounds, okay, organic compounds. They can be sugars here, okay, amino acids. They can be organic acids. You have the nucleotides, acylcarnitines, and lastly, the lipid metabolites. And they present several differences in their concentration in biological fluids. Therefore, in the current technological conjuncture, the use of more than one technique, okay, is this indispensable in order to analyze the highest proportion of metabolome, okay, as well as its concentration, its identity, and to seek an efficient construction of metabolomic profile. Now, however, the question is, which among these metabolites are actually considered part of the metabolome of the organism? It remains controversial until now because it results in partly confusing definitions. Okay? For one reason, the metabolites present in biological systems such as this, okay, they are defined as the metabolome in a very strict sense if they are endogenously synthesized, meaning they're synthesized by our body, by our cells. Okay? However, there are also metabolites that are exogenously derived. Okay, so these are what you call xenobiotics. Now, metabolites from the xenobiotics could also include the phase 1 and phase 2 metabolites, which arise from the further metabolism of the xenobiotics by various tissues and cells in our body. And the complement of these xenometabolites, including their phase 1 and phase 2 intermediates and metabolites, are what you call the xenometabolome. And this is what makes metabolomics a particularly difficult field because of differentiating between a xenobiotic or differentiating between a xenometabolome and the endogenous metabolome. However, to avoid confusion, I shall focus only on the endogenously obtained metabolites. So this is the traditional metabolomics. And perhaps you've come across this diagram highlighting the series of omics disciplines over the past couple of years, okay? And metabolomics here, if you can notice at the very bottom, is one of the omics science that is relatively new, okay? It's fairly new. It is still in its incipient stages of development here in the Philippines. I can only name a few who does metabolomics work and studies here in the country. And we are lucky that we have Dr. Lacerna as a balik scientist because she can actually provide direction to the metabolomics studies in the country. So one of the advantages of doing metabolomics is that there are only relatively few metabolites in biological system. If you compare that with the number of genes we have, as predicted by the Human Genome Project, we have about 22,000 genes, okay? While the transcriptome that arise from the genome, okay, these are mainly RNA, a complement of our RNA species, we have about 100,000 transcripts or mRNA transcripts. Now, the translation of these transcriptome or transcript leads to the complement of all the proteins in an organism, and we call this proteome. Now, the field of study that deals with the organism's proteome is what you call proteomics. And interestingly, okay, we have a lot more proteome in our body than our genes. So it is predicted by the Human Genome Project that we have 1 million proteoforms or protein isoforms, and many of which remain unidentified, okay? And however, okay, the metabolomics is actually in a very good position because due to the advancements in analytical instrumentation such as mass spectrometry and nuclear magnetic resonance spectroscopy, many of these metabolites, more than 5,000 of them, are well characterized now, okay? And interestingly, there is a subset of metabolites that capture the interest of many other researchers, such as the lipid metabolites, okay? Now, these lipid metabolites form at, around 30,000 species of them. So they are quite diverse, okay? And the study of these lipid metabolites, okay, are what you call the lipidomics. Now, compared to genomics and transcriptomics, as you can see from this figure, okay, the metabolomics can actually directly or what you say, accurately reflect the current status of the organism. And they can tell us what exactly happened to the organism because this is the ultimate manifestation of the changes in the gene expression exactly from this stage of the transcriptomics and proteomics, okay? And in other words, the inventory of all these metabolites in thousands of them in an organism can reflect the downstream activity of your genes. I'm referring to the genome. Okay, the proteins, I'm referring to the proteome, and also can offer a high level of functional description, okay, that transcends pure genomics, transcriptomics, and proteomics. That is why many researchers now are doing integrative omics, okay, in studying disease mechanisms, wherein they try to integrate their genomics, transcriptomics, and proteomics data with the metabolomics data, okay, to create a complete story of the disease mechanisms of an individual. And now it is now possible in a systems context. Dr. Lacerna mentioned the Metabo-Analyst, which is a publicly available software that you can use to do integrative omics. It is now one of the advances in metabolomics because you can do integrative approaches. You can integrate various omics data with your metabolomics data to arrive at highly functional biological interpretation of disease mechanisms. And moreover, the rapid evolution of machine learning methods, some of which have been described by Dr. Lacerna, at the same time, artificial intelligence in the last 5 to 10 years has enabled accurate analysis of these metabolites. Okay, so we can now analyze large data sets of metabolites, and this leads to an avalanche of biomedical information. If you look at the PubMed in the past 10 years, you can see that the metabolomics data are growing in numbers. There are hundreds of papers being published in metabolomics, okay, over 5 or 10 years and possibly in the next years to come because of the advancements in analytical instrumentation, as well as in machine learning or vector learning methods. Now, as I mentioned, the metabolomics target the screening of these metabolites. Okay. Now, the quantity as well as the qualitative aspect of these metabolites are very important, okay, because they appear to be very responsive to various stimuli. Okay, what are these stimuli? It is shown in this slide. This could be genetic or epigenetic stimuli. This could be environmental stimuli, okay, and also physiological stimuli such as age, okay, and other factors such as drugs, okay, nutrition, and lifestyle could also strongly influence your metabolome. And because of the sensitivity of these metabolites to various stimuli described in this slide, metabolomic disturbances have been associated with many disease mechanisms that includes cancer, diabetes, and cardiovascular diseases. So if you look at the literatures, several metabolomics actually look into applications of metabolomics, look into the disease mechanisms, targeting biomarker discovery or discovery of panel of metabolites that can actually indicate whether a disease is happening or could also be used for personalized medicine for diagnosis and prognosis of diseases. And also the metabolome is unique. It is dynamic and it is related to the phenotype of the organism. As I mentioned in the previous slide, many researchers are recognizing now metabolome as the ultimate phenotype of the individual. That's why it is now in a very good position in the era of biomedical science. Okay, and usually the ultimate goal of metabolomic is to perform high-level interpretation or biological interpretation. And this is true for untargeted or discovery metabolomics. So earlier, Dr. Lacerna discussed the approaches in metabolomics at targeted and untargeted metabolomics. Now, the untargeted metabolomics is one of the most established approach in metabolomics, especially for the purpose of biomarker discovery with applications in the clinical setting, okay? Now, this leads us to the traditional tools and strategies that are employed in metabolomics. And there are two. I think this has been mentioned by previous speakers and highlighted by Dr. Lacerna as well. So as I mentioned, I'm very lucky because I don't have to discuss this in greater detail. But I'll try to highlight some bits and pieces with emphasis on its application in the clinical setting. Okay. So first is the untargeted metabolomics. Okay, so what's untargeted metabolomics? Sometimes they call it global metabolomics because you're actually looking at all the complement of the metabolites or biochemical features in biological fluid. And most recently, scientists call them discovery metabolomics. Okay. Now, discovery metabolomics involves unbiased screening of metabolites in biological sample. I would like to highlight the word unbiased. Okay. Bakit kaya unbiased? Because you're actually using stringent data analysis to arrive at a conclusion. Okay. So you need to be able to identify these metabolites in an unbiased manner using various methodologies, analytical and also computational methodologies. And the goal, one of the goal of discovery metabolomics is to compare the patterns or profiles of metabolites among different sample groups. You may compare, for example, healthy cohorts and disease cohorts with cancer, with other non-communicable or communicable diseases. Okay, and also, discovery metabolomics involves simultaneous detection of these metabolites. That's why it is important that you use highly sensitive, highly reproducible analytical instrument for this. Okay, it involves comparing the metabolome between your experimental and control group. And you want to spot differences between their metabolite profiles. So that's actually the goal. And I think that was discussed by Dr. Lacerna as well. You do functional or pathway analysis, and eventually you validate these. Okay, so that eventually they can be applied as biomarkers and they can be validated in the clinical setting. Okay, and usually, okay, the goal of untargeted metabolomics is to perform high-level functional description or biological interpretation of these dysregulated metabolites. When you say dysregulated metabolites, I'm referring to low upregulated. Okay, those are metabolites that are above the normal levels, or the downregulated. These are the metabolites that are below the normal or physiological levels. Okay, and these dysregulation in metabolites could actually be related to you could actually relate it to a mechanism of disease, such as cancer or any other diseases for that matter. It could be communicable, okay, such as infection, or non-communicable, such as cardiovascular diseases. Okay. Now, the second approach is targeted metabolomics. Okay, so the keyword here is target because you're just looking at a specific panel of metabolites. In fact, okay, in targeted metabolomics, you're actually focusing on the validation of selected metabolites that you discovered that are dysregulated from the discovery approach. Okay, and when you say validation, as mentioned by Dr. Lacerna, you need large numbers of samples, okay? Because these large number of samples can be used to explain the effect of natural and biological variation in validating the identity of these previously identified metabolites. And I should mention that variations, either induced or uninduced, is actually rampant, okay, in metabolomics data, because metabolomics data is a multidimensional data. So by nature, this data is actually laden with a lot of variations, especially when you're dealing with individuals, okay, human population, because there are inter-individual differences, okay? Now, for that matter, you need to use large numbers of samples I highlighted here in targeted metabolomics to account for this variation. Okay, so I think that was mentioned by Dr. Lacerna as well. Okay, the induced and uninduced, making your data heteroscedastic. This is what you call the heteroscedasticity of metabolomic data. So in essence, okay, the targeted metabolomics emerges from the global screening of metabolites via the untargeted approach. Okay, and usually, if I may highlight, targeted metabolomics explore metabolites that are related to a specific pathway. For example, pathway for the metabolism of inflammatory lipids. Okay, so typically researchers would look at the various metabolites and intermediates that are involved in the metabolism of these inflammatory lipids. Okay, and oftentimes analytical and system biology approaches are used by the researcher as they look into the concentration, bioavailability, turnover, metabolism, and very importantly, the regulation of these metabolites. Okay? So, meron ka pa ring functional interpretation, high-level functional interpretation sa targeted metabolomics. It's not only routinely done in untargeted metabolomics. Now, this is the traditional workflow or the typical workflow for untargeted metabolomics. This was discussed in great detail by Dr. Lacerna. I enjoyed her talk, and I'm actually quite smiling behind the camera. Sabi ko, I don't need to discuss this in detail because we had such an excellent and brilliant presentation by Dr. Lacerna about this workflow. Nonetheless, I shall briefly describe this as it is applied in the clinical scene or setting. Okay. Now, the untargeted metabolomics typically involves these steps, okay? Profiling, okay? You have the profiling, you have the compound identification, okay, here. You have biological interpretation at the very last panel, okay? Now, I should include statistical analysis. It was discussed in detail by Dr. Lacerna. It is actually part of the chemometric analysis. Okay. Now, traditionally, chemometric analysis in metabolomics would involve this, okay, baseline correction, peak alignment, normalization, deconvolution. But more and more researchers are recognizing the importance of statistical analysis as part of the chemometric analysis. So I think it is safe to say that you can include statistical analysis as part of the profiling or chemometric analysis. Okay. Now, I will describe very briefly. As I mentioned, this was discussed in great detail by Dr. Lacerna. I really like the presentation. But just to refresh you, okay, I just quickly describe what goes on in each of these steps in untargeted metabolomics. Okay, so let's start with profiling. Okay, profiling is also known as the differential expression. Okay, so this is quite normally used in clinical medicine or clinical metabolomics, differential expression. Ano yung mga differences ng levels of these metabolites, okay? So between your experimental and control group. Now, this step involves finding metabolites, okay, with statistically significant variation between control and test sample sets. Usually, when you look at the concentration of these metabolites, you don't need to do absolute quantitation, okay? You can use relative quantitation, okay, with relative to a standard or a reference, okay? An internal standard or an external standard, okay? And the goal is actually to compile the metabolite profiles, okay? And the next step is compound identification, which until now it remains to be one of the bottleneck in metabolomics. It's very difficult to identify a metabolite if you don't have a customized or a working library. And unfortunately, this library may come at a steep cost, as mentioned by Dr. Lacerna, but fortunately, we have several databases and machine learning methods that could help us to identify these metabolites in question, okay? So the last step is biological interpretation, and which is the goal of your untargeted metabolomics, okay? When you say biological interpretation, you try to uncover the biological connections and interconnections between the metabolites, and also the result. Also, it provides some insights into the biological mechanism and the processes of diseases, as well as perturbations, biochemical perturbations in the biological system. Okay. So let me go through that very quickly. So let's start with profiling. So mabilis ko lang siyang i-discuss, okay? Because this has been discussed by Dr. Lacerna already, okay? So profiling involves finding metabolites, as I mentioned, with significant variations between your experimental and control. Okay. Now, usually in an untargeted metabolomics workflow, analytical and technical reproducibility is essential. okay, for to arrive at a stringent data analysis. And these are the keys to a successful experiment. Unfortunately, it comes with a steep price as well. If you want to have a highly reproducible, highly sensitive, and high resolution equipment, you need to invest a lot of money for these instruments, okay? And luckily, we have some of these instruments in the DLSU Central Instrumentation Facility. Okay, we have now the NMR, and it has the highest resolution in the country, and we have QTOF tandem mass spec as well, and both can be used for high resolution, okay, highly reproducible metabolite identification. Okay, now when you say high analytical reproducibility, it means that the data are the direct expression of biological variants. Now, this is very difficult in the clinical setting because of a lot of induced and uninduced variation, and at the same time, inter-individual differences. But there are a lot of approaches to neutralize this variation. And one way is via normalization, which was mentioned by Dr. Lacerna. Okay? And typically, metabolite profiling involves 4 steps. Okay? And it is shown here. Okay? Sample preparation and data acquisition, spectral pre-processing, feature extraction, and lastly is statistical analysis. Okay, so I will describe them very briefly as we go along. Okay, sample preparation and data acquisition first. This is the first step in profiling. This involves separation and detection of metabolites, and there are a lot of instrumentations you can employ, such as GCMS or LCMS or even ICMS. Now, for untargeted metabolomics, which is the focus of my presentation this morning, both the chromatographic separation and mass spectrometers must have high reproducibility to minimize analytical variation, especially when you're dealing with multitudes of samples coming from populations, okay, or epidemiological samples. Now because untargeted metabolomics aims to comprehensively catalog all the metabolites, it is imperative for the researcher that the analytical technologies perform the following functions as described here. First, it must cover a breed of metabolites with diverse physicochemical properties. So you need to first have some literature survey on the potential metabolites present in your biological sample before deciding which instrument to be used to cover as much of these metabolites in your biological fluids. Okay, so they are very diverse, as I shown you in the previous slide. They have very diverse physicochemical properties. And also, you need to consider instruments that possess large dynamic range to analyze the varying abundance of metabolites. Now, this is very important in clinical metabolomics or biomarker discovery because there are low abundance metabolites with very important biological significance, such as signaling or chemical messengers, such as diacylglycerol. They're normally present in small or minute concentrations in your biological fluid. So if you use an instrument with very small dynamic range, you will not be able to capture these important signaling metabolites. Okay, and at the same time, you also need to cover those metabolites of large abundance. Okay, what are these? These are the metabolites that are involved in energy metabolism, such as ATP, which is always abundant in biological fluids, and also intermediates, okay, that are involved in highly conserved pathways such as glycolysis, okay, and citric acid cycle. They're always high because they are actually central metabolic pathways through which many of the intermediates go through. Okay, so in fact, many of these intermediates are anaplerotic. They give rise to other intermediates that belong in a different pathway. So you see that there are interconnections between these pathways, and this is actually what you want to capture in discovery or untargeted metabolomics. That's why it is important that your platform must have large dynamic range. And this is related to the next bullet, high sensitivity to detect or quantify low abundance metabolites. I'm not going to explain that. I already mentioned that moments ago. And lastly, your instrument must possess high resolution, accurate mass detection, or capability to separate isobaric species. Now, this is true for metabolomics because many metabolites are isobaric. Okay, so in order for you to be able to detect them or identify them, you need a high resolution mass spectrometry. Okay, and usually these high resolution mass spectrometry, you normally that use orbit trap detectors with resolution uh, that reaches as high as 100, okay, 100K. They can perform this high resolution accurate mass detection or HRAM. And most of the mass spectrometers now are equipped with deconvolution programs to allow you to separate isobaric species or various metabolite species that elute at this almost the same retention time. Okay, so and also profiling involves removing background noise. Okay, this is actually one of the culprits in missed metabolite information because some metabolites have very low abundances and they tend to be obscured by these background noises. So you need to be able to separate these background noises by performing baseline correction, peak normalization, and lastly, deconvolution. All these comes very conveniently because there are now commercial softwares that can actually do this very conveniently with minimal human intervention. Okay. So this is an example of the importance of large dynamic range or high sensitivity in metabolomic platforms. Okay, I'm sure that most of you are familiar with citric acid, diba? Citric acid is an organic acid which is an intermediate in the citric acid cycle, or popularly known as the Krebs cycle or the tricarboxylic acid cycle. Now, usually, as I keep on mentioning, the requirements for the choice of analytical platform is that it must possess high sensitivity, technical reproducibility, and large dynamic range, okay, for you to have a successful metabolomic experiment. And this is particularly important in the detection and identification of low abundance metabolites such as citric acid, okay? Now, citric acid has been used. Actually, citric acid has been widely used for the establishment of metastatic cells. Okay, examples are the cells, or these are cells derived from human. I'm trying to recall human tongue squamous cell carcinoma. Okay, and different cells have different metastatic capacity, and through metabolomic screening, okay, through global metabolomic screening and eventually leading to targeted metabolomics, scientists discovered that citric acid could actually identify the low and high metastatic human squamous carcinoma cells. Okay, see, if you have an instrument with large dynamic range, you will be able to cover these very important metabolites that are of minute concentration, but otherwise very important for biological interpretation. So if you don't have a large dynamic range in your instrument, you are going to miss this important information. Imagine if you compare the citric acid across these cancer cell lines, the differences is very, very subtle. So you need to be able to capture this. You need to have a highly sensitive instrument to capture these subtle differences in metabolites, especially if you're doing targeted metabolomics. Now, another example is glutamate and glutamine, okay? Glutamate and glutamine are amino acids, diba? These are amino acids. They're considered essential and semi-essential amino acids. But other than that, glutamine and glutamate exchange is important for energy provision in certain metabolically active cells, such as kidney cells, in the event of low energy status. They could actually use glutamate and glutamine exchange pathway to extract energy from this. Okay, therefore, it is important to profile them. Okay, unfortunately, okay, glutamate and glutamine have very close structures. Okay, and they would come out at almost the same retention time And this particularly becomes difficult if you don't have a deconvolution software. You're going to miss this important information. Okay. But if you have a high resolution analyzer, such as Orbit-Chap analyzer here, you can practically separate these two, okay, metabolites, and you can effectively identify them with high level of confidence. If you have a low resolution analyzer such as QTOF, actually QTOF, personally medyo mataas pa to. The resolution could reach up to 20,000. But if you're dealing with metabolites such as glutamate and glutamine, QTOF may not be a wise choice as an analyzer to detect these metabolites. Okay, this is something to think about for the choice of platform for your metabolomics work. Okay, and lastly, feature extraction. So again, I'm very lucky. Dr. Lacerna did a great presentation on this. Okay, as if you recall, metabolomics data are highly multidimensional data. Okay, sa isang metabolite pa lang, you can actually generate a lot of information. Okay, such as retention time, mass to charge ratio from their extracted ion chromatogram or EIC. You have the intensity, you have the peak area, and so on. Let alone if you're dealing with thousands of metabolites and multitudes of samples. So your, by nature, metabolomics data is highly multidimensional. And these parameters that I mentioned a while ago are what you call the features that actually characterize your metabolites. They call that at this stage of profiling, you don't know the identity of your metabolites. Okay, the identity of metabolites, okay, can be, or the metabolites can be identified through these features. Okay, so these allow you to locate and quantify your metabolites, either relatively or absolute quantity, relative or absolute quantification. And feature extraction, again, this comes very convenient for the researchers nowadays because there are many softwares that are equipped with tick-based algorithms. They can actually do feature extraction for you, okay, simultaneously for all the potential metabolites. Okay, and kanina, Dr. Lacerna mentioned of the spectral binning. Okay, so this is part of the feature extraction as well, wherein related ions, okay, that are indicative of the extracted ion chromatographic peak, okay, are identified and they are grouped. Okay, so this is important. Okay, the spectral binning. Okay, so this is an example of feature extraction from an extracted ion chromatogram here. I am not entirely sure what metabolite this is because at this stage, we do not know yet. Okay, so that is why we need to do feature extraction beforehand. Okay, so this is the extracted ion chromatogram here. So, and then following mass spectrometry, okay, following separation, you do mass spectrometry, and then you collapse these into their mass fingerprint. The mass fingerprint then is grouped according to the ions, either singly charged, multiply charged, okay? And you do the spectral binning, okay, of these extracted ion chromatogram, okay? So these feature extraction allow you to organize your data that is now ready for subsequent analysis, okay? Now, it is very important to know that spectral binning is actually one of the acceptable data for high-level functional interpretation. So if you do this manually, it's going to take a lot of time, but there are many softwares now that can do spectral binning, which is a lot more convenient, okay? Okay, and lastly, the statistical analysis. And again, happy that Dr. Lacerna mentioned this statistical analysis. And traditionally, we do two types of statistical analysis, the univariate and multivariate. Perhaps the more familiar to you is the univariate method, such as t-test and ANOVA test. They're commonly used. However, one of the pitfalls of this univariate method is that they don't take into account the presence of interactions between the different metabolic features as if they occur independently. Okay, so this is actually not underscored in univariate methods. In fact, univariate methods is normally applied in targeted metabolomics. Okay, Now, for discovery metabolomics, the most useful is the multivariate method. Again, I enjoyed Dr. Lacerna's discussion on this, very clear, detailed, rich in information. So you can do supervised multivariate analysis, such as principal component analysis, or you can actually label your features according to classes and do supervised. Sorry, this is supposed to be, PCA is unsupervised while PLSDA is supposed to be supervised, or partial least squares determinant analysis. Okay, so as Dr. Lacerna mentioned, this allows us to, because your metabolomics data are highly multidimensional, so many parameters, so many data, so you can project them into fewer components. So you can do this beforehand to look at the differential expression of these metabolites between control and experimental group. Okay, and this is routinely done before metabolite identification. Because if you skip this step and you do metabolite identification, and after that you realize that there's no difference between your control group and disease group, in this case you have wild type and mutant, then metabolite identification will be practically pointless. That's why statistical analysis, I emphasize multivariate methods, is a very crucial step, okay, in the differential expression analysis. This gives you the proof of concept so that you can proceed to the metabolite identification and eventually do high-level functional interpretation such as pathway analysis and metabolite set enrichment analysis. Okay, so metabolite identification is our next topic. Okay, so again, I'm glad that this was covered by Dr. Lacerna. So this is actually the bottleneck on untargeted metabolomics research. Okay, even my group has had difficulties in identifying metabolites because we don't have a library. We don't have a customized library to identify these metabolites, and we mainly rely on databases. And in fact, database search forms one of the important platforms for metabolite identification. Okay, so for MS database searching, you could use Metlin or MZ Cloud. Okay, however, it lacks sufficient specificity for identity confirmation. When you want to do validation, the MS database searching may not be as useful, okay, as it sounds. But that's the first approach usually, especially if you don't have a library, which is quite costly. So you could use this mass spectrometry database searching. And then now, if you have tandem mass spectrometry, in which case DLSU-CF has it, then you can do tandem mass spectral library matching. Okay. Now, these library matching employs fragmented molecular ions. Okay, that can be compared against the tandem mass spectral libraries or electron impact libraries. Okay, and along with your retention time, okay, this can provide the high level of confidence. Okay, so now this is an example of a library. Okay, so this is a commercial library, by the way. It already comes with your, for example, with your QTOF tandem mass spec, especially if you want to dedicate that instrument for metabolite or metabolomics work. You need to have a dedicated library for your targeted metabolites. And this comes with a steep price as well. Okay, so, but it's very convenient because you don't have to use those databases anymore. So right there, you can customize it, okay, depending on the known metabolites from the previous literatures. And you can identify these metabolites with high confidence. But oftentimes, there will still be metabolites that will be left unidentified, even if you use these costly libraries, okay? So that is why you need to do other approach, okay? Especially if you're not able to identify a particular metabolite. What can be done, okay? Then you will have to rely on your knowledge on your mass spectrometry, okay? Or possibly in your NMR spectroscopy. So this requires high-level skills, okay? The de novo interpretation, where you reconstruct a chemical structure based on fragmentation data without prior knowledge on them, okay? And also you can do structural correlation. Usually, this is an in silico method comparing your experimental mass spectra, okay, from that of the in silico generated mass spectra of a particular metabolite. Okay, but this is very useful, okay, to structural correlation, and there are a number of publicly available databases that can be used for this structural correlation in the absence of these commercial softwares. Okay, one such commercial software or database is what you call the My Compound ID or MCID. Now, this is a web-based tool for metabolite identification based on integrated mass spectrometry or tandem mass spectrometry. Okay, sorry about the annotation. Now, the MCID library contains predicted tandem mass spectra. Take note, this is in silico generated tandem mass spectra of over or close to 400,000 known and potentially existing human metabolites. Okay, and the current MCID library now includes over 8,000 endogenous human metabolites that are curated in the Human Metabolome database, and almost 400,000 predicted human metabolites in EML. I forgot what EML stands for. Evidence-based Metabolome Library, or EML, which is based on one metabolic reaction only. Okay, that's why between HMDB and EML, HMDB still proves to be more useful in metabolite identification for discovery metabolomics. And the last step is functional interpretation. So I'm going to go through this quickly because this was already discussed in the previous presentation. Now, in functional interpretation, you can use a lot of public software such as Metaboanalyst. Metaboanalyst could actually give you provide you interactive graphical displays. Para siyang Google Map based graphical map. Shows you the identified metabolites and position them on pathways that help reduce their function. Okay, so it is again goes back to the HMDB. One of the databases used by Metaboanalyst is actually Human Metabolome Database, or HMDB. But other databases are also used, such as EML and also Metascience. Group of metabolites, okay, now in functional interpretation, the group of metabolites that are related to the same biological processes are placed in unique metabolic pathways following stringent statistical categorization, okay? So sometimes the software will actually set the settings by default, or you could actually even more stringent, especially if you want to do clinical validation later, you have to be quite stringent with this step. Okay, wherein you place certain metabolites in biological pathways and eventually underscore them as overrepresented or significantly dysregulated. Okay, and the purpose of this functional interpretation is to identify connections between metabolites. Okay, and that is the goal of discovery metabolomics, right? You want to find connections and interconnection between this metabolite, because they can actually provide you insights into these disease mechanisms. And eventually, you want to use this as biomarker for biomarker discovery. And when validated, okay, clinically, they can be applied in the clinical setting for diagnosis of diseases and also for assessing response to treatments or as prognostic markers. Okay, now, high-level functional interpretation usually uses these databases as outlined by Dr. Lacerna. Okay, the KEGG, or the Kyoto Encyclopedia of Genes and Genomes, the Metascience, okay, the Small Molecule Pathway Database, SMDB, the Wiki Pathways also is quite useful, but the number of pathways, okay, that are cataloged or curated in Wiki Pathways is not that much compared to KAGG. Okay. And lastly, if you're dealing with plant metabolites, you can also use PMN or Plant-Science. And this is actually a multi-species pathway database for plant metabolomics, okay, such as rice. Now, Metaboanalyst, okay, came around some years ago, and there is now, I don't know the newest version, I think version 5. It now integrates high-level omics analysis, where you can integrate data, okay, from your genomics, transcriptomics, and proteomics, along with your metabolomics, and you can obtain high-level functional description of your metabolomics work. Okay. Now, in comparison with other programs such as XCMS, Galaxy, your metabolomics, as I mentioned by Dr. Lacena, it's like a one-stop shop. You can do spectral pre-processing, you can do normalization, you can do statistical analysis, okay, both supervised and unsupervised and supervised. And I think one of the magnificent applications of these metaboanalysts is the high-level functional interpretation, such as pathway analysis and metabolite set engagement analysis, and also pathway topology analysis, which identifies the centrality of your metabolites, okay, in a given pathway. Okay, so this is an example of metabolomics data interpretation. It made use of a commercial software. This is not generated by Metaboanalyst, but this is highly intuitive because it actually project your data on a network or systems context, their connections, okay, and also the pathways they are involved. Okay, and you can also do heat map analysis so that you can actually gain insights into the relative, okay, expression of these metabolites, okay, using heat maps. Okay, now heat maps is not exclusive in metabolomics. It's been applied for many years in genomics, transcriptomics, and proteomics, but fairly recently, it is now widely applied in metabolomics as well. Okay. So I mentioned a while ago that the focus of this presentation is how can we employ metabolomics, okay, especially basic research in metabolomics in the clinical setting. Now, if I may be honest, this is actually a long way to go, okay, application of metabolomics sa clinical setting. It is because you need to validate, okay, metabolic markers, okay, in a large population because before you can actually apply it in the clinical setting. But that is highly possible, okay, in metabolomics work, but it's going to be really ambitious. But a lot of research groups now are working towards translational medicine and personalized medicine, wherein they employ metabolomics data in understanding disease mechanisms, okay, which later on can be validated for personalized medicine, okay. Now, so I would say one of the most widely used application of metabolomics is biomarker discovery, okay. I'm sure you are all familiar with biomarkers, okay? And you know what biomarkers is. By definition, biomarkers are a characteristic that is biochemical characteristic that is objectively measured and evaluated as an indicator of normal biological process or a pathophysiological or pathogenic process. And very importantly, the responses of an organism such as humans to pharmacological treatments and other environmental factors or any therapeutic or nutritional intervention. Okay? In fact, the biomarkers form the cornerstone of the treatment individualization in personal medicine. That is the goal. You need to individualize, okay, the biomarkers for the diagnostic of diseases and also assessing response to various treatments or therapeutic intervention. Okay. Now, however, okay, the application of metabolomics, okay, in the clinical setting suffers from multiple patient-specific factors. Okay, it can be these multiple patient-specific factors may be influenced by the metabolome but you can control this, okay, variation using a carefully designed research study to minimize the variation. However, there are patient-specific factors that will affect, okay, your metabolite identification and biological interpretation. So you have the inter-individual variation. Okay, for example, the demographics of your experimental group or your cohort, such as gender, age, body weight, their comorbidities, their concomitant medications, all these can actually influence the metabolome. Good thing now that we have some machine learning methods and algorithms that can actually neutralize these variations. Okay. Now, I think it is important in the clinical medicine when you apply metabolomics in the clinical setting is that the biomarkers could be dynamic or could be static biomarkers or static prognostic biomarkers. So what's the difference between dynamic and static prognostic biomarkers? Okay, so when you say dynamic markers, biomarkers, they describe the disease progression and they are associated with the disease response. Okay, so over time, they can vary. So there will be temporal differences in these biomarkers, and you need to be able to capture them over time. So this is what you call longitudinal metabolomic profiling. Okay. Now, on the other hand, the static prognostic markers, okay, these are usually used for prognostic purposes, and the aim is to predict clinical response or a dynamic biomarker thereof. Okay, so usually you start with a static biomarker discovery, and then you can proceed to dynamic biomarker discovery. Okay, now examples of dynamic biomarker discovery would be um, response to treatments, or how does chemotherapeutic intervention influences certain metabolites over time. Okay, so static prognostic biomarkers, okay, examples of this is biomarkers that could actually diagnose, okay, with high confidence certain diseases such as cancer. Okay, and recently, colleagues of mine from the University College of Dublin patented metabolite biomarkers that could actually lead to high-level diagnosis of certain types of prostate cancer among the Irish population. Now, why do we need metabolomics? Okay, why do we need this in the clinical setting? Well, we have a lot of clinical biomarkers for the diagnosis of diseases, but these biomarkers, okay, they suffer from low sensitivity at times and or lack of specificity. Okay, majority of these biomarkers have been developed or identified decades ago, and however, we still need we still need other biomarkers or indicators because many of these biomarkers are not very sensitive in disease diagnosis, okay, or even as a prognostic marker. So I'll give an example that was provided by Dr. Acerna. The creatinine and urea are traditionally used as a biomarker for kidney disorders. It's traditionally used in the clinical practice as markers for clinical kidney disorders. However, these two biomarkers, they are sometimes not very sensitive in detecting certain renal disorders, especially chronic renal disorders. Therefore, we need innovative metabolomics work, okay, which comprises molecular intermediates, okay, in the pathway, and also end products resulting from different cellular and physiological process. So this is the advantages of metabolomics because they could actually reflect dysregulation in the cellular physiology and pathophysiology relating to a disease. Okay. Now, more on precision medicine. Okay, I think you are well aware that most of the metabolomics applied in the clinical setting start with a proof of concept study. Okay, for example, in clinical metabolomics, biomarker discovery, ultimately you generate a quantitative data that are chemically and biologically interpretable, and it's of great importance in disease mechanism. Okay. Now, however, okay, one of the weaknesses of this is individuals have inter-individual variation. Okay, so therefore you need large number of samples, okay, from your population cohorts to arrive at a biomarker, metabolite biomarker that can eventually be used or validated and apply in the clinical setting. Okay, now, in fact, a large number of clinical metabolomics employ this, okay, case control studies, okay, and these case control studies are aimed at identifying differences between metabolite signatures, okay, of group of patients such as healthy and disease. Now, however, such categorization are often inappropriate or oversimplification, especially if the treatment individualization biomarkers, okay, are inherently dynamic processes. Okay, so especially if you're looking at metabolites that are by nature very dynamic or fluctuating. Okay, so that's why you need to employ a very carefully designed research when you want to employ metabolomics in the clinical setting, okay? And as I mentioned, this suffers clinical metabolomics suffers from serious intra-individual variability, and this results in a highly convoluted intra and inter-individual variation or metabolic signatures. Within the same individual, okay, there would be dynamic changes in the metabolites. Okay, that's why it's very difficult to capture them using static approach. So you need to employ both static and dynamic, okay, approach, okay, in the clinical metabolomics. So now, eventually, okay, you will arrive at metabolomic profile. Okay, that's actually one of the goals, okay, as outlined in the workflow in metabolomics. Based on this metabolomic profile, you can actually generate a model, okay, emerging from your high-level functional interpretation, such as pathway analysis, metabolite set enrichment analysis, and pathway topology analysis. And eventually, as Dr. Lacerna mentioned, you need to be able to validate this in larger patient cohorts so that eventually it can reach the clinical bench, okay, such as for the application for treatment individualization biomarker or prognostic biomarkers, such as what is known as the personalized or precision medicine. Okay. Now, however, as I mentioned, there are still a lot of things, okay, that should be done, okay, in the field of clinical metabolomics so that eventually it reaches the clinical setting. Okay. But over the past couple of years, there have been significant advancement in analytical instrumentation and systems biology that allow that actually propel these metabolomics, clinical metabolomics, leading to applications in the clinical setting. Okay. Now, these are some of the opportunities and challenges in metabolomics aside from understanding disease pathophysiology. Okay, you could also use metabolites signatures as potential biomarkers of nutritional deficiency, and that is actually one of the goals of my research group. We use biological fluids to underscore changes in metabolites in biological fluids such as serum plasma and breast milk from Filipino lactating women. Then, very interestingly, you can classify these individuals into their specific metabotypes. Okay. Now, this is good for nutritional surveillance, okay, and can be combined with your genomics data. So if you have certain genotypes that could potentially lead to a dysfunction in the metabolite makeup on an organism, then you can actually do metabotyping of individuals or population. And lastly, assessment of metabolic changes following nutritional intervention or modification, and integrative approaches, okay, in understanding disease pathophysiology that requires genomics, transcriptomics, proteomics, and metabolomic data. So it's actually across multidisciplinary approach to be able to arrive at a mature and functional, highly functional data, okay, that will describe your disease mechanism. Okay. Now, in the next few slides, I'm quite close to conclusion. In the next few slides, I will just share to you what we have been doing, okay, as part of my metabolomics initiative in De La Salle University. So this is actually in collaboration with the Food and Nutrition Research Institute of the Department of Science and Technology. As you know, FNRI is the premier arm of the government in food and nutrition research. Been lucky to collaborate with them in this study, and they provided the precious breast milk samples from the Filipino lactating women who participate in the National Nutrition Survey. So, to my knowledge, our group is the first one to characterize the comprehensive metabolome of breast milk using both NMR and mass spectrometry approaches. So this shows you representative total ion chromatogram from the GCMS analysis of the breast milk samples obtained from the FNRI. So these are the Filipino lactating women, okay? So we identified several metabolites using GCMS, and majority of them are lipid metabolites, such as glycerol, caproic acid, capric acid, lauric acids, and so on. And we also identified other metabolites, such as hormone intermediates. This is actually a work of my master's student who has now graduated. Okay, Gerald. Okay, he also do NMR analysis of the breast milk. Okay, so we actually grabbed the opportunity to use the NMR over in DLSU SCIF, a very powerful NMR, and in collaboration with UCD Conway Institute in Ireland, we were able to probe a lot of metabolites, okay, using the NMR. So, for example, we identified certain amino acids, okay, organic acids, okay, derivatives of amino acids, okay, and also lipid metabolites such as choline, phosphocholine, and their intermediates, okay. And we were able to conduct high-level functional interpretation using MetaboAnalyst. So MetaboAnalyst is a publicly available program that it's highly cited by researchers, and it's been peer-reviewed. So do not shy away from using this because it's very, very useful and very beneficial for startup researchers on metabolomics. Okay, so this is actually the metabolite pathway network overview as the result of the metabolite set enrichment analysis obtained using the MetaboAnalyst software. Okay, as you can see, there are nodes here that were identified as overrepresented, and as expected, most of them relate to lipid metabolism, such as bioacid biosynthesis, and also amino acid metabolism, and also beta oxidation of very, very long chain fatty acids. Okay, and this is the graphical overview of that breast milk metabolome, highlighting the centrality of those metabolites that we identified. Okay, so each of these nodes, okay, corresponds to the pathway impacts, as mentioned by Dr. Lacerna, of the matched metabolites of a given metabolic pathway. Not only it indicates a significantly dysregulated pathway, but also the centrality of these metabolites in this pathway. So for functional interpretation purposes, you could use pathway topology analysis in MetaboAnalyst to assess the centrality of your metabolites, okay? And also, when you say centrality, the significance of these metabolites in a particular pathway. Usually, these metabolites serve as intermediates, okay, for the synthesis of other important metabolites in a given pathway, and that highlights their functional importance in a given pathway. So this can be done in MetaboAnalyst via pathway topology analysis. So this is the summary of my presentation. So metabolomics, let me reiterate that metabolomics, its main goal is to screen small molecules that we call metabolites in complex biological samples or biological fluids. It provides a snapshot of the metabolism This is also called the fingerprint, biochemical fingerprint of an organism. And one of the advantages is that this can reflect the physiological state of the organism, understand the, or reveal information on disease phenotypes. And among the omics approaches, I highlighted metabolomics being very sensitive to various environmental and physiological stimuli. Therefore, many researchers now are recognizing the importance of metabolomics, serving as the ultimate phenotype of organisms, including humans. In clinical metabolomics, which aims to identify these small molecules present in patient-derived samples, such as blood, serum, okay, serum, plasma, urine, sweat, saliva, breast milk, okay, they've been attracting attention to support discovery of novel disease biomarkers. However, it remains to be a bottleneck, okay, in its application in the clinical setting. In fact, consensus in best practices for the integration of metabolomics in the clinical practice remains to be warranted or are still lacking at this stage. Okay, but as I mentioned, in the years to come, there are maraming advancement, not only in analytical instrumentation, but also in systems biology that could actually address this limitation of application metabolomics in the clinical scene. Lastly, I'd like to thank everybody who attended this webinar. It's very well attended. I'm quite nervous presenting a while ago, realizing that there's over 100 attendees. So congratulations to the organizers, the DLSU CIF, PCHRD DOST, Dr. Camacho, Dr. Lacerna. Thank you for inviting me. I'm very delighted. Although I must say I'm not yet established in metabolomics, I'm a very early researcher. I'm at a very early career stage as a metabolomics researcher. So at this stage, I don't consider myself as an established person or established metabolomics scientist, but I'm really enjoying investigating a lot of disease phenotypes using metabolomics platform. So maybe over the years, with the help of my colleagues, especially Dr. Lacerna, we will be able to establish metabolomics facility in the university, and who knows, it could be a resource center here in the country, especially that metabolomics is still in the incipient stages of development in the Philippines. Thank you again and have a nice day. I'll be delighted to answer questions during the open forum.